Hello, we're recording here from the Rolling School of Public Health at Emory University. This is the Virulent Vortex, a podcast in which we discuss infectious diseases, from molecules and pathogens to populations, pandemics, everything in between. My name is Jaap de Rode. I'm a professor of biology here at Emory and today's host of this show. And with me is
Erin Scherer. I'm an assistant professor in the Division of Infectious Diseases at Emory University School of Medicine.
Well, Erin, thanks for being on the show. And I'd love to hear about your research. And my understanding is that you work on papillomaviruses and vaccines. So maybe as a little bit of introduction, can you explain to us what papillomavirus is and why you are interested in it?
Yes, of course. Thank you for having me, first of all. Human papillomaviruses are DNA viruses that infect humans through typically sexual contact, and they can infect the ones we're interested in, I should say. They can also infect through non-sexual contact. So if you've ever had warts, those are also human papillomaviruses, but they're not the ones that we're going to be focusing on today. So the ones we're focusing on today are spread through sexual contact. They infect mucosal surfaces, so not the skin on your hand. And they are known, at least 12 types are known to cause cancer. So that's like cervical cancer, anal cancer, vaginal, vulvar, penile cancer, and oropharyngeal cancer, which is really on the rise, particularly in men.
So you mentioned human papillomaviruses, you mentioned cancer, but you also mentioned the genital warts that some people may be more familiar with. So how common are these viruses? And so, for example, are you studying these viruses here in the United States or in different places? And how common, who gets them? How old are people when they get them?
So, great question. So this virus, the human papillomavirus, is the most sexually transmitted virus of any sexually transmitted pathogen. So essentially, if you've ever had sex, you will probably get human papillomavirus. And we see typically two peaks. One, when people are in their teenage years. When they are first having sex for the first time in their life. So whether that's late teens, early 20s, but we typically see a peak then. And then we see another peak when folks reach middle age for whatever reason. Some think that's potential changes in lifestyle. So people may be separating at that point and dating again or getting remarried, or it may be that that's when our immune system starts to kind of wind down, and so we may be seeing infections crop up again. But what I would say is that that you asked about is that so there's the age, there's the fact that it's very prevalent, but also, as I said, the types that don't cause cancer, like the types with your skin, most people have also been infected with those types.
Yeah. Okay. So they're very common viruses.
They're extremely common.
And so how common is the cancer versus, say, the warts?
So luckily, I can at least say that for the cancer, most people have been infected, but less than 1% will go on to get cancer. And it's actually like not just infection, but you have to have infection, and then you have to have what we call persistent infection. So infection that continues for like 6 to 12 months, you're not getting rid of it. So if we continue to test you with swabs, it'll show up as DNA positive, right? Because that's the virus. And then essentially after you see that prolonged infection, you'll start to undergo these initial stages where we see cells that are growing abnormally. And then it takes, at each of those stages, you could clear the infection, you could stop, you could not progress to cancer. So again, at each stage, like fewer and fewer people might be the ones that progress to full invasive cancer.
Yeah. And so this is then also where the vaccines come in. So you're working on vaccines. And so the idea is you can vaccinate people so they don't get the warts or they don't progress to the cancer? What is the idea of the vaccine?
Yeah, so the idea is that the vaccine does not clear the virus. It does not clear the cancer or the genital warts. And so the goal is to vaccinate people before they're infected. And so for this reason, it's recommended for the ages of 9 to 26. It's approved for those ages 9 to 45. And I also should point out, because I get a lot of questions about this, but actually, if you get the vaccine between the age of 9 to 14, it's only recommended to get 2 doses. because the antibody responses are really great before you hit puberty. At actually 15 and older, even if you get 2 doses, they'll be slightly lower than what we see in that younger age group. So it's recommended because you'll have a better immune response the earlier you get it.
Yeah, so it's really preventing. And so then you mentioned that the vaccine you're working on is based on proteins. So, you say some vaccines are based on inactivating the live virus. Others are based on RNA molecules, for example, the SARS-CoV-2 virus. Yeah. So this one is a protein one. And what is the benefit of having that protein?
Well, the benefit of having a protein versus like a live attenuated virus is that more people can take that vaccine. So if you're a pregnant person or if you're immunocompromised, you might not be able to take a live attenuated vaccine, but you would be able to take a protein-based subunit vaccine.
And that's because the live attenuated could actually become a real infection.
Yes, it does. It does basically have a weaker infection, and sometimes that can get out of control. Yep. And then the other thing to say about the virus-like particles, the protein base, is they really mimic exactly what the virus looks like.
Yeah. Okay. Yeah. And so your work specifically, do you design the vaccine? Do you try it in humans, in animals? What is going on in your lab?
Yeah. So we have, I'll tell you first what question is that we're looking at and how we're addressing it. So the first question that we're trying to ask is how does this vaccine generate such durable antibody responses? And so we're doing that both in animal models and mice, and we're also doing that in humans. And so I'll tell you the story about mice first. So a few years ago, in the early 2010s, the FDA reported that there was some DNA in one of the HPV vaccines, and it's not thought to cause any safety concerns, but it's really residual from the manufacturing process. And so we know that HPV, as a double-stranded DNA virus, it encapsulates its genome, which is, double-stranded DNA inside of that L1, that virus-like particle. And so we sort of thought maybe it's taking up this DNA part of the manufacturing process, and maybe that DNA is causing an adjuvant effect. And what I mean by that is maybe it's really responsible for either boosting these high levels of antibodies or making sure they're really durable. We don't know which.
Yeah.
So one of the questions we're asking, because we can't knock out genes in humans, it's not ethical, not allowed, is that we're basically immunizing mice with the same vaccine that humans receive. And we're doing that in wild type mice or regular healthy mice. And then we're also administering this vaccine to mice that lack pathways in the immune system that sense that DNA that activate immune responses. And so that's sort of what we're looking at.
And so that allows you to look at the protein on its own versus the protein plus the DNA.
Yes.
And so with adjuvant, you mean something that actually strengthens the immune response?
Yes.
Because that's generally used in vaccines, right?
Yes, correct. And actually the HPV vaccines, a few of them have adjuvants. There are actually more than one being used, and I'm not going to get into it, but they have an additional adjuvant that are very common in vaccines.
And so that's the mice, but you also mentioned humans.
Yes. So really, the most important model is the human model, right? The most relevant. And so we have a clinical trial that's ongoing in healthy adults 18 to 45 years of age. And there we're really trying to understand what types of immune responses this vaccine is generating. And we're paying particular attention to a few different things. So one, the antibodies that you find in the blood are generated by very special cells that are found in your bone marrow, long-lived plasma cells. So we're interested, is there something special about those cells versus other vaccines? So we can look at like the genes they express and see if those are different somehow and they're particularly long-lived. We're also looking in lymph nodes, so where they very early after vaccination to see if there's something special about the way those B cells are activated to become these long-lived plasma cells that secrete this antibody. And we can then, try to see if there's some, unique gene signatures that are occurring. And also, no one's really looked at this vaccine that in depth before. And so we can then take this initial study and compare it to other studies. For example, now there's a call for having a one-dose HPV vaccine. So we can ask, how does a one-dose HPV vaccine in humans differ from this traditional 3-dose vaccine in those that are older?
Yeah. And so what, with that in mind, your mice and your humans, what do you hope this research will do for protecting humans?
Yeah, so that's a great question. And the reason why we're really interested in these long-lived antibody responses is if we can figure out how the vaccine is doing that, then we hope to apply it to others. So, for example, we have confirmed that there is DNA present in these vaccine lots, and we had immunized those mice that are knocked out in DNA sensing, and we've also immunized the wild-type mice. And so far, we don't see any difference sort of like right after the full vaccine series. So at the peak of the antibody response where we expect them to be highest. But now it's really exciting because we're now at a year with these mice. And now we have these specimens, we're testing them right now in the lab, and we're really excited to see if now there's a difference later on. So is this DNA somehow helping activate B cells that turn into long-lived plasma cells better than if it wasn't present? And if that's the case, then, for example, we might think about making new protein vaccines with DNA-based adjuvants.
I see. So not just for the human papillomaviruses, but potentially other ones as well.
Yes, absolutely.
Yeah. And then maybe as a final question, so when we talk about these vaccines, clearly they can be protective, and your work is really figuring out, what is the best way to make that vaccine, when to administer, how many doses, what gives us the best immune responses. Are you also looking at any potential negative effects of these vaccines? I know there is a lot of worry with people and vaccines. Is that something you're looking at too?
Well, to be honest with you, the HPV vaccine has an extremely safe safety profile. And the initial concern was really around fainting. So they were seeing a lot of people fainting after they received the vaccine. And then as time has gone on, that more data has been collected, and they realized that it's because this vaccine was being given to adolescents. And adolescents actually, as a general group, have higher incidence of fainting. And so actually, if you give it to older people, you don't see the same results. So I would say I agree that there are some vaccines, and there's reasons why we're developing new ones, because they may have some side effects. But the HPV vaccine, it's a very safe vaccine. It's a very effective vaccine. And it's one of the few vaccines that I really encourage people to take, because many of these vaccines, they protect against up to 7 types that cause cancer. So you might have been infected by one type, but you might not have been infected by the other 6 types. So if you're eligible to receive the vaccine, in my opinion, and again, I'm not an MD or a PhD, but I think it's better than not.
Yeah. So even if you already have had some infection with some types of the virus, you can still get the vaccine to protect against other ones.
Yes, that's correct.
Wonderful. All right. Well, it was really great talking to you. Thank you for sharing your exciting research.
Thank you. Thank you for the invitation.